So let's suppose, let's take an example. Here, a Salmonella typhimurium TA102. So this is a strain of Salmonella. There are strains of E. coli also where we can test these chemicals, and those are tryptophan, operon. There are mutation at tryptophan, in the tryptophan operon, and where we see a tryptophan negative to become a tryptophan positive. It is same. We are talking about Salmonella typhimurium TA10 strain of Salmonella carries the his G428 nonsense mutation. So his is, in this case, his is his. I'm talking about histidine operon, and I'm talking about nonsense mutation. Okay, a mutation which completely, nonsense mutations are those mutations which completely change the type of amino acid which they code, which encodes, so at this, so this strain, so which encodes an okra of stop codon. So TA is a stop codon. Why I'm saying it's a nonsense, it's a nonsense mutation. In place of stop codon, now it will code for glutamine, so which is CAA. So what you have to notice is that this T has been substituted with C, and because of this single base pair substitution, a stop codon, which a codon which was meant to stop the translation, has now been turned to a glutamine coding codon. So this will be called as nonsense mutation. Okay, because this mutation is making no sense. Something which the codon which was meant for stopping the translation has been turned to a glutamine coding codon. And similarly, there are other type of missense mutations which you can look. And if you want more on this, I can make a separate class on this. So what happens that at position 847 of histidine operon strain, so strain TA10 can reward the histidine prototrophy by base substitution at either the Ocre site in histidine or extragenic Ocre suppressor sites, anticodon change of tRNA genes. So reversion at the Ocre site, that is TAA, include 5 types of mutation. So 5 types of mutation can occur, like that T can be replaced with the C. Okay, very well, it's okay, because we want that stop codon at 50th position to be removed. I'm taking that example again. So let's suppose this stop codon is at the 50th position, and because of this, my histidine coding enzyme, the enzyme which will help in the histidine biosynthesis, is no more functional. So I want to replace this T in a way that, so that it start coding for some amino acids. So let's suppose this T has changed to C. Similarly, this A can be replaced with this T, and so on. You can look into this. Among them, 7 possible sense mutation induced by single base substitution. Okay, so similarly, there are other strains also where a proline triplet CCC has been replaced with leucine triplet at position 434. So what the take home message is that I am trying to convert a strain which is oxotropic, which require histidine in its growth media so that it can grow, because there is a mutation in the histidine operon of this strain, and by themselves they are not able to synthesize histidine, so they are dependent on external histidine. Now if I apply a potential chemical which can change this mutation to a functional codon, so that a functional peptide or a polypeptide or a enzyme can form, so that they can make their own histidine. Now they get into a prototrophy state. They are not dependent on external histidine. They can synthesize their own histidine. So that is all we are going to talk about. So there is a paper. There are many papers. You can look into this paper if you want more understanding about this. So what's the procedure? Isolate an oxotropic strain of Salmonella typhimurium, and let's suppose this is TA102 strain of typhimurium. This is one TA10, and we know that there is a stop codon. TA, prepare a test suspension of histidine negative Salmonella typhimurium in a plain buffer with test chemical. Let's suppose in this chemical is fluorine. Now I want to test that this chemical aminofluorine, I want to test that whether it is a potential mutagen or not. So add small amount of histidine, because a small amount of histidine allows the bacteria to grow for an initial time and have a chance to mutate. So what is the line is trying to tell you, that when you take a histidine salmonella bacteria in a suspension, you must add some histidine, because some histidine, not too much of histidine. I'm adding small quantity of histidine so that this bacteria can grow. If this bacteria will not grow, then how you are going to test the chemical? So first we will grow a bacteria in a way that a minimal amount of histidine will be given so that this bacteria can grow. Now there is no more histidine. Because whatever amount of histidine was there, it has been consumed by salmonella. Now there is no more histidine. So if my chemical, the test chemical, which is 2-aminofluorine, is a potential mutagen, if it can convert this histidine auxotrophy, because there is no more histidine now in medium, if this test chemical is not a potential mutagen, what will happen, that this salmonella typhimurium is not going to grow further, because whatever histidine which was there in medium has been already been consumed. Now all histidine has been consumed. So and this chemical is not a mutagen, so this will not convert this histidine negative to histidine positive. But if at all this test chemical, which is 2-aminofluorine, has changed the histidine negative to histidine positive, we know that already the whole histidine has been consumed. If this mutagen changes the histidine negative to histidine positive, then they will grow. And if it is not, then they will not grow. And if it is not a potential mutagen, then they will not grow. And if it is a potential mutagen, then they will grow. Third, also prepare a control suspension of histidine negative cell. So you very well know what is the why control suspensions are important, because in control suspensions you are not going to add chemical. So where you will see that once all the histidine which was present in the media, the minimal amount of histidine that you gave, which was once it will get used, then bacteria are not going to survive. Okay, so that's a control. And incubate the suspension at 37 degree for 20 minutes. Now inoculate the suspension on culture plate lacking histidine. We know why, because if the compound has changed the histidine, if the compound has changed the histidine negative to histidine positive, so by themselves they can synthesize their own histidine and they can grow. And incubate the plates at 37 degree for 48 hours. After 48 hours, count the number of colonies in each plate. The mutagenicity of chemicals is proportional to the number of colonies absorbed. If large number of colonies on test plate is observed in comparison to control, then such chemicals are said to be mutagens. Very few number of colonies can be seen on control plates also. These represent the spontaneous point mutations on histidine encoding gene. So let's suppose what I am telling so what I am trying to say, that if you get 1 or 2 colonies or 3 colonies in control plate, it is okay, because we know that the bacteria can incorporate some mutation during time of replication. Okay, so at the time of replication, few mutation can be incorporated which can turn the histidine negative to histidine positive. So 2-3 colonies are normal. But whereas in case of the test, there will be more number of colonies if the compound is a potential mutagen.